Millions of families struggle with the progressive cognitive decline of Alzheimer's disease, fueling an urgent global push for a cure. This pressure culminated in the FDA approval of monoclonal antibodies like lecanemab and donanemab. These intravenously administered therapies are engineered to target and clear extracellular amyloid-beta plaques from the brain parenchyma. This chart shows the gap between biological and clinical results. While these drugs can clear nearly 90%, this framework models the disease as an extracellular phenomenon, where toxic amyloid plaques aggregate in the spaces between neurons and spread damage toward the cell. Dr. Gunnar Gouras, a 2022 Oscar Fisher Prize laureate, advocates for a model known as the inside-out paradigm. Gouras identifies Alzheimer's as an intracellular trafficking disorder that begins within the neuron's own endosomal-lysosomal system. In this paradigm, the extracellular plaque represents the necrotic debris left behind after a neuron destroys itself from within. This internal focus targets the specific biological events that precede the loss of the synapse. We zoom down the extreme length of a single axon to reach the distal synaptic terminal. Because synapses are located at massive distances from the cell's main degradative machinery in the soma, they must rely on local synaptic endosomes to recycle proteins. This diagram shows the initiation of the disease at the synapse. On the left, amyloid precursor protein, or APP, is located at the plasma membrane. The cell internalizes APP from the surface into the early endosome through a process called endocytosis. The acidic environment of the endosome allows the base 1 enzyme to cleave APP, generating the amyloid-beta 42 peptide. This hydrophobic amyloid-beta 42 accumulates on the outer membranes of multivesicular bodies, blocking the local recycling system. This biochemical failure occurs entirely within the synapse, shielded from drugs that operate only in the extracellular fluid. This internal blockade intersects directly with the primary genetic risk factor for the disease, the ApoE4 allele. Under normal conditions, the ApoE protein acts as a vehicle to deliver essential lipids and cholesterol into the neuron. As the diagram indicates, the E4 variant suffers from impaired recycling. Once internalized, it becomes pathologically trapped within the endosomal network. The trapped ApoE4 encounters the accumulating amyloid beta within the same constricted endosomal space. This convergence triggers endosomal enlargement and a total failure of the synaptic recycling needed for memory consolidation. The intersection explains why the presence of the ApoE4 gene significantly accelerates the early stages of synaptic failure. As these internal aggregates build, they physically disrupt the microtubule architecture that supports Beyonce. The neurite swells into a bulbous, structurally unstable mass, a dystrophic neurite, severing the electrical signals required for memory. These swollen structures were first documented in 1907 by Oscar Fisher, whose observations of neuritic dystrophy remained marginalized by the field for a century. Modern high-resolution immuno-electron microscopy provides the visual proof of this massive internal swelling that Fisher's early drawings predicted. In the final phase, the synaptic membrane reaches a breaking point. It cannot contain the mass of indigestible aggregates and undergoes a violent rupture. Necrotic lysis creates the extracellular plaque, marking a final morphological stage of the disease. This paradigm explains the limited efficacy of monoclonal antibodies like lecanemab. These large Y-shaped molecules are confined to the extracellular space. They cannot cross the cell membrane to reach the amyloid beta actively aggregating within the synaptic endosomes. While these drugs neutralize the extruded plaques, they fail to rescue the neurons currently suffocating from internal toxicity. These antibodies also target amyloid that has become structurally integrated into the walls of the brain's blood vessels. Stripping this amyloid compromises vascular integrity, leading to the brain bleeds and edema known as ARIA. Aggressive clearance of end-stage plaques causes severe collateral damage while leaving the active intracellular disease intact. Halting neurodegeneration requires therapeutics capable of penetrating the neuronal membrane. One priority involves developing small molecules to restore the acidic pH of the lysosomal machinery, preventing the initial buildup of waste. A second target involves engineering drugs to correct the aberrant retention of ApoE4, allowing lipids to flow freely through the endosome once more. A third strategy seeks to activate internal proteases to lower the amyloid beta burden before the neuron crosses the threshold of apoptosis. Gouras's research provides a modern molecular basis for the cellular swellings Oscar Fisher documented over a century ago. Curing Alzheimer's requires shifting resources from the inert plaque toward the preservation of the living synapse.